Alzheimer's disease is a complex tapestry of molecular malfunctions and cellular catastrophes that culminates in the loss of memory, identity, and autonomy. Today, we explore the scientific and medical intricacies of this devastating neurodegenerative disorder. First identified by Dr. Alois Alzheimer in 1906, Alzheimer's disease is the most common cause of dementia, characterized by progressive cognitive decline and neuronal loss. It is a multifactorial disorder involving genetic, environmental, and lifestyle factors. Central to its pathology are two hallmark proteins known as amyloid-beta and tau. As of recent estimates, over 55 million people worldwide are living with dementia, with Alzheimer's disease accounting for approximately 60 to 70% of cases. The incidence doubles every 5 years after the age of 65. By the year 2050, projections suggest this number could surpass 150 million due to aging populations. Risk factors extend beyond age and genetics to include hypertension, diabetes, obesity, and sedentary lifestyles. What occurs at the molecular and cellular level during Alzheimer's disease? What defines the pathophysiology of this condition? On a molecular scale, Alzheimer's disease is marked by the abnormal processing of amyloid precursor protein, or APTP. Typically, this protein is cleaved by alpha secretase. However, in Alzheimer's, APP is improperly cleaved by beta secretase and gamma secretase complexes. This results in the production of neurotoxic amyloid-beta peptides, particularly a form known as amyloid-beta 42. These peptides aggregate into oligomers and fibrils, forming extracellular amyloid plaques. These amyloid plaques disrupt synaptic function. Simultaneously, tau proteins, which normally stabilize microtubules in neurons, become hyperphosphorylated due to dysregulated kinase and phosphatase activities. Hyperphosphorylated tau detaches from microtubules, aggregating into intracellular neurofibrillary tangles, leading to cytoskeletal collapse and neuronal death. Chronic neuroinflammation also plays a crucial role in Alzheimer's disease. Microglia and astrocytes, which have immune regulatory roles in the brain, become activated by amyloid-beta deposits, releasing pro-inflammatory cytokines and reactive oxygen species. These factors in turn exacerbate inflammation and neuronal damage. Additionally, mitochondrial dysfunction and impaired autophagy further contribute to the disease's progression. Autophagy is a crucial intracellular process that normally clears dysfunctional mitochondria and toxic proteins, such as amyloid-beta and tau. Impaired autophagy can therefore lead to a buildup of amyloid-beta plaques and tau fibrils, which leads to further neurodegeneration. Impaired autophagy is therefore thought to contribute to Alzheimer's disease. Clinically, Alzheimer's disease manifests with an insidious onset of memory impairment, particularly in forming new episodic memories due to dysfunction in a brain region known as the hippocampus. As the disease advances, deficits emerge in executive functions, visuospatial abilities, and language, known as aphasia. Further cumulative deficits emerge, including the brain's ability to control and perform complex movements and tasks, known as apraxia, and diminished abilities of recognition, termed agnosia. Neuropsychiatric symptoms, including apathy, depression, agitation, and psychosis, often complicate the clinical picture. Standardized cognitive assessments, such as the Mini Mental State Examination (MMSE) and the Montreal Cognitive Assessment (MOCA), aid in quantifying cognitive deficits. Advanced neuropsychological testing can delineate specific domain impairments. Diagnosis of Alzheimer's disease integrates clinical evaluation with biomarker evidence. The National Institute on Aging and Alzheimer's Association (NIAAA) provides research criteria emphasizing biomarkers. Cerebrospinal fluid analysis reveals decreased amyloid-beta 42 levels and increased total tau and phosphorylated tau proteins. Imaging modalities include MRI, which reveals cortical atrophy, particularly in the medial temporal lobes and hippocampus. FDG PET demonstrates hypermetabolism in the parietal and temporal lobes. Amyloid PET imaging utilizes radiotracers like Pittsburgh compound B to visualize amyloid deposition. Emerging blood-based biomarkers measuring plasma amyloid-beta 42 to amyloid-beta 40 ratios and phosphorylated tau offer less invasive diagnostic options. Current pharmacotherapy aims to mitigate symptoms and slow progression. Cholinesterase inhibitors such as donepezil, rivastigmine and galantamine inhibit acetylcholinesterase, increasing acetylcholine availability at synapses, thereby enhancing cholinergic transmission, which is deficient in Alzheimer's. The NMDA receptor antagonist memantine blocks the pathological overactivation of NMDA receptors by glutamate, thereby reducing excitotoxicity. Recently, monoclonal antibodies targeting amyloid beta, such as aducanumab, have gained attention. Aducanumab purportedly facilitates the clearance of amyloid plaques, though its clinical efficacy and approval have been subjects of debate. Non-pharmacological interventions focus on cognitive rehabilitation, behavioral therapies, and lifestyle modifications to support neuronal health. The relentless progression of Alzheimer's imposes a profound burden on patients and caregivers. Behavioral and psychological symptoms, such as aggression, wandering and sleep disturbances, strain familial relationships and necessitate comprehensive care strategies. Caregivers often experience chronic stress, depression, and financial hardships. Multidisciplinary support, respite care, and counseling are essential components in managing the disease's holistic impact. While definitive prevention remains elusive, epidemiological studies suggest that modifiable risk factors play a significant role. Strategies include cardiovascular health. Managing hypertension, hyperlipidemia, and diabetes reduces vascular contributions to cognitive decline. Lifestyle interventions. Regular physical activity enhances neurogenesis and synaptic plasticity. Cognitive engagement. Through education, bilingualism, and mentally stimulating activities, cognitive engagement builds cognitive reserve. Dietary factors. Diets rich in antioxidants and anti-inflammatory components like the Mediterranean diet may confer neuroprotection. Social engagement. Social interactions stimulate neural networks, potentially delaying symptom onset. Genetic counseling for individuals with familial Alzheimer's, particularly those with mutations in APP, PSEN1, and PSEN2 genes, is also pertinent. Advancements in molecular biology and genomics propel Alzheimer's research toward novel therapeutics. For example, beta-secretase inhibitors aim to reduce amyloid beta production by inhibiting the initial cleavage of APP. Tau aggregation inhibitors target the accumulation of hyperphosphorylated tau to prevent tangle formation. Immunotherapy involves both passive monoclonal antibody therapy and active vaccine immunotherapies, which are in trials to enhance the clearance of amyloid beta and tau. Gene therapy techniques like CRISPR-Cas9 offer potential to correct the genetic mutations associated with early onset Alzheimer's. Targeting neuroregeneration through stem cell research is exploring the potential replacement of lost neurons and the restoration of neural networks. Finally, systems biology approaches and artificial intelligence are being employed to understand disease complexity and identify therapeutic targets. Alzheimer's disease is one of our greatest biomedical challenges. As we unravel its molecular mysteries, each discovery brings us closer to effective interventions. Through rigorous science, collaborative research, and clinical trials, we strive to illuminate a path forward for those affected. Thank you for joining us on this comprehensive journey into the depths of Alzheimer's disease. If this exploration enriched your understanding, please like, share, and subscribe to Science Curious.